All right, it is 3:03 Central Time. Great to see everybody. My name is Jason Vogel, the director of the Great Plains Stormwater Center of Excellence. We're excited to have our two guest speakers today, Jacoby King and Madeline Henderson. But before we get to their talk, especially for those of you that are new to our webinar series, just wanted to share a little bit about the Great Plains Stormwater Center of Excellence. So the University of Oklahoma is one of, and the Great Plains Stormwater Center of Excellence is one of 4 centers of excellence in the U.S., all funded by the U.S. EPA over the last year to year and a half. The Southwest Center of Excellence is led by the University of Nevada. The Cold Climate Center of Excellence is led by the University of New Hampshire and the University of Minnesota. And the Southeast Coastal Center of Excellence is led by the Center for Watershed Protection in collaboration with numerous university partners. So for our center, we kind of have 2 different parts. One is research and one is outreach. But for our research projects, we do things like green stormwater infrastructure forensics, where we investigate established systems and go out and do inspections and experimentation on them, working in the area of smart stormwater, and also developing a web-based tool for green stormwater infrastructure feasibility, specifically focused on smaller rural communities in our region. We're also developing a couple of white papers on extreme event adaptation for stormwater practices and also on innovative funding approaches. On the outreach side, we have a lot of different activities. You guys are here for our webinar every month, and we'll rotate through topics and also do virtual open houses. We also provide technical assistance throughout the region for individuals, for cities, for tribes, for states, whoever might need some assistance related to stormwater management, especially as it comes to green stormwater infrastructure in places where it hasn't been implemented before, but really any stormwater project topic is on the table. We also have targeted rural and tribal outreach. Save the date, the Great Plains Stormwater Conference will be coming to Oklahoma City next spring, March 27th to the 31st. We're working with the NASA Globe Observer to establish a citizen science project. Be on the lookout for some more with that. We're also establishing a green stormwater infrastructure atlas. And finally, we're establishing a peer communities network that will be rolled out within the next month. So thank you very much. Here's our website, gpstormwater.org. You can use that QR code to find us. But with that, I'm going to turn it over to our speakers, Madeline and Jacoby. If you guys want to share your screen and get started.
Yeah.
Should be sharing right now. Well, loading at least.
There
Yes.
we go.
There we go.
See? Wonderful.
Perfect.
All right. Hello, everyone. Thank you for joining today. My name is Madeline Henderson, and this is Jacoby King. We are presenting to you from the University of Oklahoma, and we're both research associates at the Oklahoma Water Survey. I have a background in microbiology and a master's of public health and epidemiology, which shapes my lens and expertise contributed to this project. And Jacoby brings to the table a master's of microbiology and extensive experience in molecular biology as one of the longest tenured members of our team. Today, we'll be presenting some project results for microbial source tracking, which use qPCR-based host-associated markers. This project took place in Tulsa, Oklahoma, which is the northeast part of the state, between May and November of 2025. We'd like to take a moment before we get started to introduce our wonderful team. So the sample analysis was carried out here at the University of Oklahoma. We'd like to shout out our amazing leadership, including Dr. Jason Vogel, Dr. Kristen Shelton, and Dr. Bhagia Galkazadewage. We are thankful for Diana Macias, who led the sample collection and coordination with the City of Tulsa. We would also like to acknowledge Dr. Cody Watley for his contributions to the data analysis for this project. This project was funded by the City of Tulsa, and we'd like to thank their team as well. Thank you, Skylar Robson, Brian Lewis, Lauren Ireland, and Jessica Sims. We are thankful to have the opportunity to collaborate with such a fantastic team. We wouldn't be able to present these results without the help of all of these folks. Thank you so much to everyone for contributing hours of their time and expertise to this project. So we have a lot to cover today. We'll be starting with a brief background and project introduction before going over our methods. We have combined our results and discussion sections today, which cover 3 separate analyses performed using the results captured in this project. Finally, we will end with a discussion of data actionability and recommendations, as well as where we hope future research will take us with microbial source tracking. So the ultimate goal and objective of this project was to address fecal contamination in environmental waters. This fecal contamination can be serious and potentially deadly, with approximately 90 million illnesses each year related to microbial exposure in U.S. recreational waters. These are diarrheal illnesses most often, things like E. coli, Shika toxin-producing E. coli, norovirus, cryptosporidium, and Giardia. Furthermore, a lack of regulatory compliance prevents recreational use and creates a substantial financial burden in the form of healthcare costs and remediation. Section 303D of the Clean Water Act, as outlined by the EPA, requires states, territories, and tribes to identify bodies of water that do not meet these water quality standards. The agencies that facilitate this monitoring in Oklahoma include the Oklahoma Water Resource Board, Oklahoma Department of Environmental Quality, and many other local partners. Bacteria is one of the leading pollutants in Oklahoma and causes impairment in streams. These streams that consistently fail to meet water quality standards for E. coli or fecal coliforms are required to have total maximum daily loads, or TMDLs, developed. So where does this bacterial and fecal contamination come from? Well, fecal contamination sources can look different depending on the location and the land use surrounding the recreational body of water. Some examples of fecal contamination sources may include leaking septic systems and combined sewage overflow to local wildlife and agricultural practices. This can also include things like improper pet waste disposal and migratory animal populations. This figure from Martin 2024 illustrates some of these pathways from combined sewage overflow and agricultural landscape. It's effectively describing how stormwater drives bacteria impairments by mobilizing runoff that contaminates water bodies, exceeding regulatory thresholds, and elevating that public health risk. This creates the need for a tool that goes beyond just determining that there's fecal contamination present, but also provides insight on how to remediate and prevent this contamination, which is where microbial source tracking comes in. Microbial source tracking, or as we will continue to refer to it as MST, is a powerful tool because it allows for the identification of fecal contamination sources in recreational waters. This is facilitated through the detection of microbes unique to hosts of interest like human, livestock, or avian species. I think this is a really nicely illustrated image from Vade et al. 2025, showing the diverse range of hosts from urban and rural environments that are all contributing to one shared body of water. This really emphasizes the need for protection of water quality through the investigation of things like land use, rainfall, and geology.
Yeah. So for this project, in collaboration with the City of Tulsa, we were able to design several assays to detect a pretty wide range of hosts that might contribute to fecal contamination in recreational waters. One important consideration with MST is assay specificity. So we always want to make sure that our fecal markers correctly identify the targets of interest without unintentional cross-reaction with other species. To prevent this, we use only literature-validated assays for our targets. We landed on 5 assays, including human, ruminant, dog, general avian, and geese. As you can see, targets can be general or species specific. The selection of an avian assay and goose assay is something that can better illustrate species specific contamination. This can help develop more tailored intervention if a host is, say, like consistently detected in the samples. One other thing that I really want to bring up is that these assays were developed for Northeast Oklahoma, where there's a mixture of urban and rural space. However, one of the advantages to microbial source tracking is that it can be customized to the hosts and sampling locations of interest. So this brings us to our sampling sites chosen for this project by the City of Tulsa folks. There were 31 sampling locations, all of which were 303 delisted for the year 2024. The impairments for these streams included Enterococcus, E. coli, and turbidity. These sample sites were split up into 3 groups, including watershed characterization sites, you can see here in red, of which there are 7. Zinc lake sites, you can see in blue, of which there are 4, and then TMDL sites, of which there were 20, and obviously makes up the lion's share of our samples. So quality assurance and quality control measures were implemented throughout this project. We included field replicate sampling for each of these sites specifically. Sampling rotated through the sites, and samples were collected weekly, again between May and November of 2025. Samples were collected by the City of Tulsa employees and then retrieved by an Oklahoma Water Survey courier who immediately delivered the samples to our lab at the University of Oklahoma. The time frame for processing bacterial samples is sensitive, so we allow a maximum of 48 hours from time of collection. Our lab processed these samples immediately upon reception. So the samples were vacuum filtered through a 0.45 micron membrane filter. This setup is pictured here on the left. A DNA extraction was performed on the filter and accompanying biomass using a kitless extraction method. This is something that uses a lysis buffer followed by magnetic beads, which bind to the nucleic acid content, allowing for like organic and inorganic material to be removed through successive wash stages. The extraction of DNA from environmental samples is pretty notoriously difficult at times. So especially in samples where like sediment and other inhibitors are present. So in order for us to understand how well the extraction was done on these samples, we run an additional assay called Sketa. Sketa is salmon sperm DNA, which behaves similarly to the DNA which we're extracting, but notably, it's not something you would expect to find in Oklahoma streams, right? So we spike a known concentration of Sketa during the extraction process, and then compare that to the amount found in each sample via qPCR to establish a recovery percentage for each sample. We're happy to report that over the duration of this project, we achieved a 98% sample pass rate, so pretty good. Okay, as alluded to, following extraction, samples underwent molecular analysis via qPCR, or quantitative polymerase chain reaction. If you're not familiar with qPCR, this is a molecular method that uses specific primers and probes, which are short strands of DNA, which pair with and amplify our gene targets of interest. The number of relevant gene copies is quantified via fluorescence, which is picked up obviously by a computer, a detector. For each qPCR assay that we run, we also run a standard curve, which allows the machine to compare the fluorescence of the synthetic DNA to that of the samples and allows for the titular quantification of the amount of DNA present. So one thing to note, for added confidence in our results, we also plate each sample in technical triplicate, just as a means of quality control. Okay, so notably, this project generated a lot of results, which leads us to our statistical analysis portion here. The primary analytical data set contains 866 observations drawn from 1,011 raw measurements across 7 markers, 32 tributary sites, and 36 sampling dates. So heavy duty stuff. So some of these things to note about the data which were generated by this project. Failed recovery samples were excluded as they would introduce unquantifiable error to the analysis, so better just be without. Likewise, field duplicates were reserved only for QA/QC analysis. Now I've got some acronyms for you here. So non-detect, ND, and detected non-quantifiable results, DNQ results, are left-censored observations, i.e. the true concentrations lie somewhere between 0 and the limit of quantification, the LOQ, but can't be precisely determined. So rather than excluding these observations entirely, which biases summary statistics upward and reduces sample sizes, censored values were substituted for concentration-based analyses. For ND samples, these were handled by substituting the value with the limit of quantification divided by 2. For DNQ samples, where the replicate values were above the limit of quantification, the geometric mean of the replicates is used as the concentration estimate. And then for DNQ samples with replicate values at or below the limit of quantification, these are treated as censored and assigned that same sort of value as the ND samples, the limit of quantification divided by 2.
So for our initial data set, we looked into the detection frequency, which is an indicator of a persistent issue within the sample locations, suggesting repeat detections throughout the entire season. Detection frequency was compared across sampling groups by marker. We also looked at the concentration of the markers, and we stratified these into quartiles to assess the impact of the markers by site. This allowed us to have a greater picture of which sites were the most problematic contributors of fecal contamination. We also organized this in upstream to downstream trends, which allowed us to visualize where contamination may have been introduced. So for that detection frequency, we found that the avian marker was detected 100% for the entire season. We were expecting this to be detected broadly, but we weren't necessarily expecting 100%. But then when looking specifically at our Canadian geese, we see that fecal contamination was most frequently detected in those zinc lake sites, which sit along a heavily developed part of the Arkansas River. Goose contamination was not tested for in the watershed characterization sites, as you notice the absence of that bar. The human markers were also detected most frequently across sites. You can see an 80 to 90% detection across all of our sampling groups. And the ruminant markers showed moderate detection frequency for the recreational season, again in those zinc lake sites. And then lastly, we have our dog marker for contamination. This had the lowest detection frequency, but was notably high, around 26% in those TMDL sites. Here are a few tables that we built to show the stratified concentration by site and marker. We know a lot of folks joining today are not from the area, so we won't dwell on this for too long, but we'd like to point out some of the trends that we found interesting. You notice that these TMDL sites generally exhibit elevated concentrations for all of the markers, whereas our watershed characterization sites, particularly Berry Hill Creek and Douglas Creek, had higher quantiles compared to other sites like Salt Creek and Upper Mingo Creek. And then lastly, we have our zinc lake sites at this top table, which showed that elevated ruminant and avian concentration. And then, of course, we also organized these into those downstream trends. I would like to make a quick note. So for our TMDLs, those are split up along 4 different streams. So they're characterized or organized by their flow into or within the Arkansas River. And also our watershed characterization sites, of course, encompass a broad geographic area. So those Berry Hill and our Salt Creek are much further spaced than these. But you can see some trends of increased and decreased concentration for this is the human marker. Notably in Hakey Creek, you can see an increase in concentration as you move downstream, whereas within some of these watershed characterization sites, you can see a notable decrease in concentration as they move downstream, which we found very interesting. So our initial results suggested that the responsible hosts and sample locations it suggested responsible hosts and sample locations with persistent or elevated concentration, which indicated potential locations for implementation of best management practices and interventions. However, there are several elements that facilitate fecal contamination, kind of going back to the background and introduction. And we believe these results are meant to be interpreted alongside other data for meaningful next steps.
So in this next section, we're going to cover 3 additional analyses that we performed to investigate factors associated with fecal contamination, like E. coli concentration and those that might mobilize fecal contamination in environmental waters like rainfall and land cover or land use. Elevated E. coli concentrations in surface waters serve as a primary regulatory indicator for fecal contamination and associated public health risk. The EPA's guidelines established the 10% threshold value at 406 most probable number, a benchmark that generally indicates water is unsafe for recreational use. Although E. coli can't be used alone to determine or remediate fecal contamination sources, we wanted to investigate the association between concentrations of E. coli and concentrations of host biomarkers. An analysis was designed using E. coli concentration collected by the city of Tulsa in our MST dataset. A Fisher's exact test was conducted to assess whether the MST marker detection was more likely to occur when E. coli levels exceeded thresholds set by the EPA. And then a censored Kendall's towel was employed to assess the correlation between reported E. coli concentration and MST markers. This is useful because it can compare variables that may include D and Q data, which again is common in environmental samples. It's something that doesn't require you to assume a linear relationship. So here you can see the censored Kendall's tau, and as you can see, there is evidence of a significant relationship between E. coli concentration and human, dog, and avian markers. For the analysis looking at marker detection and concentration during E. coli exceedances, we found that concentration of human and dog biomarkers were significantly associated with E. coli concentrations above 406, most probable number. So these results, they help inform the relationship between E. coli concentration and MST results to establish a baseline understanding for potential contamination sources. The results indicate that E. coli exceedances are significantly associated with dog and human markers, including indicating rather that potential hosts might be these for contamination events. However, this is something that should be used in conjunction with microbial source tracking efforts. Understanding the relationship between E. coli levels and MST markers is essential for identifying pollution sources, prioritizing management interventions, and informing regulatory decision making.
So we also looked at the relationship between our data set and the rain events. So as we know, precipitation events represent significant drivers of hydrological and water quality in environmental waters. Rainfall can mobilize contaminants through surface runoff and overwhelm aging sewage infrastructure, leading to elevated levels of fecal indicator bacteria. We designed an analysis that examines the correlation between short-term precipitation events and results of the 2025 recreational season, aiming to characterize how rainfall influences the detection and abundance of markers. So for this analysis, our wet weather conditions were defined as anything greater than or equal to 0.1 inches of cumulative precipitation within 72 hours of sampling. We used a Mann-Whitney U-test for significance to analyze the relationship between marker concentration and wet-dry conditions as a binary. And then lastly, we also used a Spearman correlation to look at the correlation between the amount of precipitation and the concentration of these markers. This figure illustrates the difference in marker concentration between the previously described wet-dry binary. As you can see, there's an increase within the human and ruminant markers between wet and dry conditions, where an increase was observed after precipitation. There's evidence of a significant relationship between rain events and human and ruminant marker concentrations. As for that correlation between the amount of precipitation and the concentration of our biomarkers, we found a positive correlation and a borderline p-value for the amount of precipitation and the human marker. So overall, these findings demonstrate a significant increase in human and ruminant marker concentrations during 72-hour precipitation events. This confirms the substantial impact of rainfall on microbial contamination in the selected sampling locations. The observed elevations in human markers during rain events suggest that precipitation mobilizes sewage-related contamination. Similarly, significant increases in ruminant markers indicate that rainfall enhances the transport of livestock-associated contamination via surface runoff from agricultural areas and pastures. The potential dose-response relationship between precipitation amount and the human marker concentration indicates rainfall intensity, could influence the magnitude of sewage-related contamination events. We'd like to note that this data set was small due to limited rainfall events. Therefore, more data should be collected to further understand the relationship between rainfall and fecal contamination sources. But ultimately, we feel these results emphasize the importance of considering weather events and water quality monitoring and informing best management practices for urban stormwater and infrastructure.
Yeah. So another thing to consider for interpretation of MST data is nearby land use, excuse me. I probably don't have to tell you guys that urbanization and land use patterns are among the most significant drivers of water quality degradation in watersheds. As impervious surfaces replace natural vegetation, stormwater runoff increases in both volume and velocity, carrying pollutants like directly to nearby streams with little to no opportunity for natural filtration. As you can see on this map, sampling locations in this area encompass a broad gradient of land use conditions from heavily urbanized to forested agricultural or just other rural landscapes. So a Spearman rank correlation was performed to investigate the relationship between these land classifications and MST marker concentration. As you can see here, there is evidence of a significant positive correlation between the developed land and dog marker, this DG3 marker here. The human marker, HF183, was moderately correlated with developed and impervious settings, you can see. And then the ruminant marker here on the right, REM2BAC, it was significantly correlated with forested, agricultural, and wetland in LCD classifications. So the correlation analysis reveals this clear urban-rural divide in fecal source dynamics. Human and dog contamination are most strongly associated with developed and impervious landscapes, while ruminant contamination is what you see the most in rural, vegetated, and agricultural watersheds. Meanwhile, of course, the avian marker, it appears largely ubiquitous across all land uses. These patterns, they're consistent with ecological expectations for each source, and they reinforce the importance of land use context when designing fecal source management strategies.
Right, so ultimately, our results allowed us to identify which hosts are likely responsible for fecal contamination, and by combining microbial source tracking markers with information about sampling locations, land use, and rainfall, we're able to design best management practices. Best management practices must target bacterial reduction with required monitoring and adaptive management. This requires TMDL sites to reduce stormwater pollutant loads, demonstrating compliance through monitoring and best management practice performance. So using our results and criteria for these BMPs, we are able to develop some recommendations. So we organize these into 3 different categories by priorities. So our top priority would be just continued monitoring of all 3 sites, especially those TMDL sites based on their results of being elevated across all markers and sites, as well as TMDL designated BMP parameters. Our results would allow us to identify TMDL sites where interventions would be especially impactful, and the continued monitoring would allow those changes to be measurable. We would be able to see the impact of those interventions. We'd also recommend the continued monitoring of the zinc lake sites as it falls within this, as they're located along the Arkansas River TMDL. We would recommend continued monitoring of these sites for avian and ruminant markers, which had elevated concentrations at these sites. We'd also, of course, recommend to continue monitoring of watershed characterization sites, as microbial source tracking is able to distinguish between these urban and rural contamination sources. Our next set of recommendations are for future monitoring and interventions that would help provide more insight into these impairments. So to further understand the impact of precipitation and runoff events on the samples, we would recommend targeted storm sampling. We recommend that these are collected for all TMDL sites, as stormwater is heavily associated with elevated bacterial loads that characterize these TMDLs. We'd also collect some targeted storm samples from zinc lake and watershed characterization samples to visualize the impact stormwater has across all of these urban and rural landscapes within the watershed. We'd also like to employ more QA/QC and have recommended some challenge sampling from known fecal sources to assess the potential cross-reactivity across our assays. And then, as we previously discussed, that avian marker was detected, it had a 100% detection frequency across all of our sites, indicating that consistent widespread bird signal. But because of this, we'd like to increase monitoring of more specific markers, such as those targeting geese or other avian species alongside the general avian marker. This would ultimately help us refine our understanding of which bird species are contributing to contamination and allow us to develop more tailored recommendations for interventions. We'd also like to include additional wildlife markers and watershed characterization sites, especially those that did not have the elevated concentrations for existing markers, despite having impairments for turbidity and Enterococcus E. coli. Our last recommendation is related to future work that we believe would be invaluable to characterizing microbial contamination in impaired bodies of water and developing stronger recommendations. We'd recommend a one-time synoptic shotgun eDNA survey. This would differ from our current methods as it does not require a targeted approach. This would allow us to detect all of the fecal sources without any prior host assumptions, identifying hosts that are not captured by our targeted assays. The survey would provide insight into our current assay selections and give a more holistic picture of all of the microbial communities that are present in the samples. But overall, these combined recommendations would improve both the resolution and the reliability of microbial source tracking results, leading to more targeted and effective management decisions.
So we've got some final takeaways for you all, and what we want to impart with you is that MST is a really powerful tool for the investigation of contamination sources in waterways. It's a process which offers adaptability based on the needs of a community and its stakeholders, no matter the relevant host organisms or sampling locations. However, it's not a tool to be used in a vacuum. It should be used in conjunction with data such as E. coli concentrations, inclement weather data, and information on urbanization or land use to allow for maximum insight when planning interventions or designing future practices to prevent fecal contamination in environmental waters.
All right, thank you everyone so much for your time. We will go ahead and open up the floor for questions now.
All right.
Hey, Brandon, I see your question there, Brandon Streets. Thank you. You mentioned that the recommendations are not related to best management practices, and that is that for at this time we're finishing up the report for the city of Tulsa, and there will be some of those recommendations in there, but we have not finalized those yet or talked through them with the city of Tulsa, so we have not presented them on this presentation. But you're right, source control, best management practices are going to be very important.
Thank you, Dr. Vogel.
Yeah.
All right, other questions?
Yeah. So Rob Zet asked, did you consider deer versus sheep as ruminant sources, and why not analyze for cow? So we looked for ruminant specifically because it was something that uh, looked for all of the ruminants, which might qualify for that. It's definitely cow is definitely something that there's a ton of literature out there for it. Deer and sheep as well. I've seen the sequences for those. That's something that if folks were curious about those species specifically, that's definitely something that we'd love to do. But the people who chose these assays specifically were the city of Tulsa. So they're the ones who get to choose all the fun stuff, all the fun tests. But we got a lot of them. We could test for a lot of different host organisms.
Yeah, and I would like to note that Rum2Bac assay that we use does encompass all bovine and ovine sources. I don't believe it captures deer, if I'm remembering right.
I'd have to look.
Yeah.
I can send you the paper though, definitely, if you'd like. Feel free to reach out. So Abby Carruthers says, did you have any issues with getting amplifications when doing qPCR? If so, what changes did you make?
So given that we used all literature validated assays, this kind of gave us a head start of not having to troubleshoot things like our primer concentrations or our thermocycling conditions. So fortunately, we did not run into really any issues with getting amplification. We also did do a couple test runs with our dog assay by collecting a known fecal source and testing that to ensure that we did get amplification. But if we did run into any trouble, we would go back and, look at literature, look at our thermocycling conditions, depending on what issue it was, kind of addressing that.
Yeah. We also make use of things like dilution in order to get around the known inhibitory compounds, which are present in soil samples or anything that's adjacent, right? Like stream
Right.
water.
Yeah.
We got a couple questions in the Q&A. I'll read them to you. Sandy asked, did high turbidity impact the percent recoverability of certain markers? Utah is running into problems with high salts in our stream and recovering equine markers.
Okay. So we do use a 1:8 dilution of our sample. So we elute that our final elution is a 1:2, which I feel helps. We did have a lot of trouble with sediment in our samples, but we found that 1:8 dilution was effective in overcoming that inhibition that we saw.
And Andrew asked a question about one of a source you said 90 million annual waterborne illnesses, and Andrew notes that's huge, more than a quarter of the population. What was the source of that estimate?
I do have the source. I will have to look it back up. I don't have it on this presentation currently. Will you please send me an email and I will get that to you?
Okay, and then back in the chat, Brad asked, the Minnesota Pollution Control Agency began down the MST road a little while ago. Did you happen to consult with them or any other state or researchers on your sampling protocols?
I can answer that one. Specifically on the sampling protocols, the city of Tulsa has been doing microbial source tracking with a different group for many years. And so the sampling protocols were a continuation of the sampling protocols that they had already been doing. I do not know how many other folks they've talked with over the years as they've developed their sampling protocols. So we did not specifically talk about sampling protocols because that was prescribed to us from the city of Tulsa. And to some extent, it's sampling at their regulatory sites. And so they're relatively prescriptive on that. And then we sampled partners. We analyzed partner samples from that.
And I think you touched on this early in the talk, but James asked, are any of the elevated E. coli Are any of the elevated E. coli be contributed by septic tanks and drain fields leaching into the waterways?
Could you repeat that one more time for me, please?
Are any of the elevated E. coli contributed by septic tanks and drain fields leaching into the waterway?
So that is something that is a possibility, and that is one of the goals with MST is to look into that potential source. From our results, we can't say directly that that was what we saw, but I will let Dr. Vogel also comment if he'd like.
There are septic fields in many of these drainage areas, and so that is a high possibility that there may be some leaky septic systems in some of these areas. But we can't say for sure on any specific sample.
Thank you, Brandon, for your comment.
Yeah, that's helpful.
Okay, Andrew asked another question. Did you attribute reduction in human contaminant moving downstream in some watersheds to changing land use or development intensity?
That's certainly a factor. To be completely honest, we didn't look that in depth, but that would be something that's very worthwhile considering, in addition to things like dilution.
To some extent, in a one season's worth of sampling, I think we probably don't quite have a large enough sample set for us to make definitive conclusions on that as well. We can show what the data is doing over this one period, but I don't know if we can make those definitive conclusions yet.
So this, let's see, Julie White, thank you for your question. Have there been public infrastructure repairs that have significantly reduced fecal contamination based on your monitoring? So kind of going off of the last answer as well, this was just within one recreational season for 2025. So we can't say whether or not those public infrastructure repairs have significantly reduced fecal contamination. But if there were interventions made, future monitoring that we perform would be able to.
To add on to Madeline's answer, we are just getting ready to submit the final report to them. And then, you know, obviously they would have to act on that after they get the final report. So you guys they've seen these results in a presentation format before we present it to y'all, but this is really kind of as we're pushing the data out, we're getting it out to everybody else as well. So there hasn't been any infrastructure based upon these because the data is just getting finalized interpretation. They had the data as they went along, but infrastructure fixes don't quite happen that fast in the city of Tulsa, at least.
I don't see any more questions in the Q&A. Is there anyone in the chat have any more questions for our guest speakers?
Right. Madeline, I'm going to go ahead and share my screen quick if you want to stop sharing theirs, and we'll show everybody our website one last time. Once again, I want to thank everybody for joining us today. Thank you, Madeline, Jacoby, for a wonderful presentation. Very thought-provoking, lots of great questions. If you'd like to learn more about the Great Plains Stormwater Center of Excellence, please visit our website at gpstormwater.org, or click on that, scan that QR code that's right there, and it'll take you to the website. So once again, thank you very much, everybody. Appreciate it.